Hello class, today we're going to talk about the characteristics of skeletal muscle fibers. We're more or less going to get into the structure of skeletal muscle cells. I know last video we talked about kind of circles within circles within circles of groups of muscle surrounded by different connective tissues. We're going to continue on that a little bit more today. We, however, are first going to back up and look at skeletal muscle cells. Keep in mind, skeletal muscle cells as a whole are going to be surrounded by our paramecium. Sorry, not our paramecium. You're all correcting me, I hope. Our endomecium. Okay. All right. And sometimes we call these muscle cells muscle fibers. Okay. They are very long. They all run parallel together. You can see here they develop through fusion of myoblasts. Just break that word apart. I know it's new. Myo referring to muscle. Blast referring to build, just like we saw last chapter with skeletal system, dealing with osteoblasts. These cells become very large, they become very long, very elongated eventually over time through fusion of these myoblasts, putting them all together as you'll see on this next slide. And therefore, when they contain each of these nuclei and they fuse together, you end up with something that is multinucleate. Let's look at the picture here. You can see we start with myoblasts, muscle builder cells up here. They come together over time. Myosatellite cells, cells that help and repair those stem cells of muscle fibers. Eventually they fuse and they form something that we've seen before. Okay, this long parallel fiber. It has striations. It has many nuclei. And eventually you get what we call a mature muscle fiber. One cool side note here is that these can grow up to 30 centimeters in length. Keep in mind, you basically have, the length of a muscle is going to be from where it attaches at a tendon to a bone to the other opposite end of that muscle, where it also turns into a tendon and attaches to bone. Okay. So next thing we're going to talk about here, we're really going to get into the structures of skeletal muscle fibers. So the first couple of things we're going to talk about are some new terms definitely for you. Lots of new terms today, being the sarcolemma and transverse tubules, which we'll also call T-tubules. Okay. The reason we introduce these first is because you can see they surround and they're going to be directly involved with this large grouping of skeletal muscle fibers. So this thing as a whole is going to be a muscle fiber or a muscle cell. Now you can see there are smaller things inside of the muscle fiber. We will get to that eventually today as well. All right, but starting first with the cell membrane of a muscle fiber. We will commonly call these fibers, but do keep in mind we are talking about a cell, and all cells have a cell membrane. In the case of muscle fibers, we call that the sarcolemma. Okay. All right, and what we have then inside of the cell, instead of having just cytoplasm, we have what we call sarcoplasm. Okay, so sarcolemma referring to the cell membrane surrounding a muscle fiber, and the sarcoplasm surrounding being the cytoplasm of a muscle fiber. Now, one cool thing about the sarcolemma, thus leading into the transverse tubules, as you'll see, is that we are going to be really essentially talking about the endings of a nerve. So imagine a nerve coming in here. All right, I'm going to draw it poorly, I know. All right. And they're going to reach out and they're going to form these attachments. They're going to sit on top of this muscle fiber. So this is a nerve attaching to a muscle. And what we know is that nerves send what we call electrical signals. Okay. We'll talk about how nerves pass that onto a muscle in a later video. But for right now, just keep in mind that electrical signal is going to be passed down the length of the nerve, out into the very end, and then it's going to be passed onto the sarcolemma. All right. From there, the sarcolemma is going to transmit that action potential, as we'll call it, all right, inside the muscle fibers, all right, through the muscle fiber to all these individual tiny pieces, which we'll later talk about, called myofibrils. So to help pass that inside of the muscle fiber, we have what we call transverse tubules, okay, also abbreviated T-tubules. Keep in mind, transverse is referring to the fact that they're going to be running perpendicular to the length of the muscle fiber. The big job of the transverse tubules is to transmit action potentials from the sarcolemma inside the muscle fiber. Okay, and this is going to be what allows the muscle fiber to contract simultaneously. You don't want just part of your muscle to contract. Okay, you want just a little bit. You want to be able to contract your muscle as much or as little as possible, and this structure, these T-tubules, are going to allow you to do that, and that's where they have the same properties as the sarcolemma. Okay, so looking at the picture down here, all right, we're looking at a muscle fiber running the length here. Okay, but you can see that the sarcolemma, which is on the surface here, all right, eventually turns inward, perpendicular to the direction the fiber is running. All right, and that is what's going to allow that action potential, which travels along the sarcolemma from the nerve, to move inward, okay, inside of that muscle fiber to all those myofibrils. Okay, now let's talk about the next structure. We're going to be looking at this blue structure here in this picture, okay, right here, and it looks kind of like a lacy sleeve, and they're going to cover all of the myofibrils. All right, that lacy sleeve, as your book will call it, is called the sarcoplasmic reticulum. You may remember the endoplasmic reticulum that you talked about back in a biology class. Well, this is the sarcoplasmic reticulum, abbreviated SR, as you can see here. It's just a membranous structure surrounding each myofibril, and this is also going to help transmit the action potential to all these myofibrils. Okay, keep in mind, these smaller circles I'm circling right here are myofibrils. We haven't gotten to looking at those very much yet. All right, we're starting to right now. All right, these are all myofibrils, and altogether, these myofibrils make up this large group here, this large cell, which is going to be our muscle fiber. Okay, so muscle fiber is made up of myofibrils. What's helping move that electrical message from the nerve to the sarcolemma to the transverse tubules and now to the sarcoplasmic reticulum are all of these structures simultaneously. Okay, it's very similar in structure to the endoplasmic reticulum, as I said, and it forms chambers on the end. And you can look right here. These yellow lines are our T-tubules. On either side of our T-tubules, okay, we have kind of these blue groupings of sarcoplasmic reticulum. Those blue groupings, let's see here and here, all right, are what we call terminal cisternae. Okay, looking at that term there, another new term. Okay, they're attached to the T-tubules. They're butted up right against it. So whatever action potential the T-tubule has is then going to pass it to these terminal cisternae. Keep in mind, terminal means end, and cisternae means something kind of like a cistern, something that is going to hold something very special. And we'll talk more about what that is down the road here. Okay, all right. Together, the T-tubule and two terminal cisternae. Okay, keep that in mind. The terminal cisternae that occur on either side. So here's a T-tubule, and you have 1, 2 terminal cisternae on either side. That makes 3 things. Well, we have a triad here. Okay, that's the term for it. Okay, now the cool part is T-tubule sends an action potential. That action potential travels into the terminal cisternae, where we have the largest concentration of calcium ions stored in resting, note resting, muscle. Okay, when the terminal cisternae are stimulated by the action potential from a T-tubule, they're going to release calcium into what we call the sarcomeres. Okay, sarcomeres are just a small part. They're not a circle within a circle now. They're a small part of what we call our myofibrils. All right, and we're going to talk about the sarcomere here a little bit more in another video. Okay, but it's that release of calcium ions from the terminal cisternae into the sarcomeres that begins muscle contraction. And ultimately, that's what we're trying to get at this entire chapter. How do we contract a muscle? Okay, all right, we're going to get much more specific with some of these things, but we really got to talk about these structures first before we can talk about what's going on with them. Okay, now looking at this picture, a lot of this is new terms that we should have seen now today for the most part. We look at the outside of this entire thing. Keep in mind, this whole picture here is a picture of a muscle fiber. Okay, and a muscle fiber is also a cell. All cells have a cell membrane. That cell membrane here is called the sarcolemma. All cells also have cytoplasm inside of them. All right, a jelly-like substance in there, which everything is housed in. In the case of a muscle fiber, we have a sarcoplasm. Okay. Now there is something smaller inside of a myofibril. Sorry, get ahead of myself. A muscle fiber, and they are called myofibrils, which we'll end up talking about more here in a second. Okay. All these individual circles here and here, and this whole thing sticking out over here. That's sticking out. Those are myofibrils. Our last, I promise, circle within a circle. Okay. Now you can see the sarcolemma covers the outside of the muscle fiber. It's all this here. Okay. And at various points, we have this yellow structure here, which penetrates and is perpendicular to the direction the fiber is running. That is going to be our T-tubules. Okay. Whatever action potential the sarcolemma has is going to be passed on to the T-tubules, which then is going to pass it to the myofibrils via the terminal cisternae. And you can see our sarcoplasmic reticulum along with the T-tubules on either side. You can see these groupings of sarcoplasmic reticulum. That's where all the Ca2 plus is stored. Okay. All right. When that action potential gets passed inward, all right, that's going to cause calcium ions to be released into our sarcomere. Okay, and we're not going to get too much into sarcomeres yet in this video. We'll have a whole separate video on those. All right. Now, last bit I want to talk about are the myofibrils themselves, the last circle within a circle. This is a lengthwise subdivision of a muscle fiber. It's made up of the protein filaments. We talked about those protein filaments at the very beginning of the chapters. There are two types of protein filaments, the thin filaments and then the thick filaments. Okay. Now, if you think about this a little bit, what do we know about skeletal muscle fibers and skeletal muscle as a whole? Is they are striated. Where do those striations come from? They come from alternating thick and thin filaments throughout the skeletal muscle fiber. So looking at this picture here, you can see we have a dark band. That dark band is going to be made up of our myosin. Okay, they are the thick filaments. Keep that in mind. We also have light bands, that lighter pink in there. The light bands are going to be made up of actin, and the actin then therefore makes up the thin filaments. So when we're talking about thick and thin and light and dark, we're talking about what can pass through in terms of light. So looking ahead here for a second, you can see in this picture, this is what we call a light micrograph. This is where we're passing light through a small section of muscle. All right, and you can see we have these bands. We have thin filaments contained over here in this section, and then we have thick filaments in this section. When we pass light through them, more light is able to pass through the thin than is able to pass through the thick. So they appear striated when you look at them using light. Okay? All right, so that's an important question you're going to have to be able to answer later. Now, the last thing I want to mention is that we're going to get much more specific with actin and myosin and their structures in the following videos, and therefore, once we get down all the way to the small molecular level, okay, we're finally going to be able to talk about how muscle contracts. Thanks, and I hope you enjoyed.